Welcome everyone. In the previous lecture, we started nitrogen cycle, right? So, nitrogen cycle has 5 basic steps. What are the 5 basic steps of nitrogen cycle? Come on, tell me. What is the first step called? It is called nitrogen fixation. This is the first step of the cycle. And the second step will be nitrification. Right, children? And the third step will be nitrogen utilization or nitrogen assimilation is the third step. And the fourth step will be ammonification. And the last step will be denitrification. Right, children, these are the 5 steps of the nitrogen cycle. What are the 5 steps of nitrogen cycle? The first step is nitrogen fixation. Then conversion of ammonia to nitrite and nitrate is called nitrification. After that, the plant will absorb that nitrite and nitrate, and then it will make amino acids, proteins, chlorophyll, all the substances and all is called nitrogen assimilation. Then afterwards, when plant dies, animal dies, the complex proteins are broken down and ammonia is released. That is called ammonification. The last step where the soil nitrogen gets converted into atmospheric nitrogen. From the bottom, the nitrogen going up is called as denitrification. In the previous lecture, we started with the first topic called nitrogen fixation. In nitrogen fixation, we discussed that there are 3 types of nitrogen fixation: industrial nitrogen fixation, electrical nitrogen fixation, biological nitrogen fixation. Now, in biological nitrogen fixation, we were talking about symbiotic nitrogen fixation between Leguminaceae plant and Rhizobium bacteria. Rhizobium leguminosarum is the name of the bacteria which will do nitrogen fixation in Leguminaceae plants, right? There we were talking about diazotrophy. So our today's topic will be diazotrophy, children. Now come on, what is diazotrophy? Diazotrophy can also be called as nitrogen fixation. This is nothing but nitrogen fixation. Diazotropy is nothing but nitrogen fixation. What is nitrogen fixation? Nitrogen fixation is nothing but conversion of N2 to ammonia. Conversion of N2, nitrogen gas, to ammonia. Reduction of nitrogen to ammonia is called nitrogen fixation. We were talking about the bacteroids, right? Bacteroids, which are active stages, they synthesize an enzyme called nitrogenase, which will convert the nitrogen to ammonia, right? So we were talking about the Rhizobium bacteria. The Rhizobium bacteria will synthesize an enzyme. What is that enzyme will synthesize? Rhizobium bacteria synthesizes nitrogenase enzyme. It synthesizes nitrogenase enzyme which will catalyze nitrogen to ammonia. But nitrogenase enzyme is sensitive to oxygen. It will not work under aerobic conditions. So who will come and remove the oxygen so that anaerobic conditions are maintained, so that this enzyme converts nitrogen to ammonia? Leg hemoglobin. Rhizobium bacteria synthesizes nitrogenase enzyme which is sensitive to oxygen. The nitrogenase enzyme is sensitive to oxygen. So, leg hemoglobin acts as a scavenger. It acts as a scavenger. It traps the oxygen. Now, cyanobacteria like Frankia also will do nitrogen fixation. Now, in Frankia, then leg hemoglobin is not there. Then who will remove that oxygen? In cyanobacteria, example nostoc. In nostoc, there are certain structures called heterocysts. In cyanobacteria, heterocyst will protect the nitrogenase enzyme from oxygen. In bacteria, leg hemoglobin will protect the nitrogenase enzyme from oxygen. In cyanobacteria like nostoc, so heterocysts protect the nitrogenase enzyme. They protect the nitrogenase enzyme from oxygen. So in rhizobium, leg hemoglobin will take care of oxygen, whereas in cyanobacteria, heterocyst will take care of oxygen, so that they ensure that anaerobic conditions are maintained and the enzyme can simply do its function. Now let us purely talk about enzyme in this class. What is the name of the enzyme, children? It is nitrogenase enzyme. Let us clearly see what is this nitrogenase. Nitrogenase is an hexamer, means made up of 6 subunits, and it has 2 components. Nitrogenase enzyme has 2 components. The first one is called component 2, I will write first, and the second one will be called component 1. Component 2 is a dimer made up of 2 subunits, whereas component 1 is a tetramer made up of 4 subunits. 4 plus 2, 6 children. That is why nitrogenase is called hexamer. Now, what are the 2 units which make up the component 2? They are 2 iron molecules. Since component 2 is made up of iron, component 2 is also called as iron protein. It contains iron protein. 2 iron molecules will be there. Component 1, it is tetramer. It is made up of iron molybdenum. One iron molybdenum, another iron molybdenum. So it is called tetramer. So it is called iron molybdenum protein. This is called only iron protein. That is called iron molybdenum protein. And this is a part of an enzyme. You can also call it as dinitrogen reductase enzyme. The component 2 of nitrogenase enzyme is also called dinitrogen reductase enzyme, whereas component 1 can be called as dinitrogenase. Component 1 is called as dinitrogenase. So we are dividing the nitrogenase enzyme and we are telling that nitrogenase enzyme has 2 components, component 2 and component 1. Component 2 is made up of 2 subunits, so that's why it's called dimer. Whereas component 1 is made up of 4 subunits, that's why it's called tetramer. What are these 2 subunits? 2 iron only, 1 iron, another iron. So what are these 4 subunits? Iron, molybdenum, iron, molybdenum. So here 4 are there, here 2 are there. This iron protein is also called dinitrogen reductase enzyme, and iron molybdenum protein is also called dinitrogenase enzyme. And this component 2 works with ATP hydrolysis. It needs energy to do its function. Component 2, it works with ATP hydrolysis. Means for the component 2 to act, it will hydrolyze ATP. Whereas component 1, it doesn't require any energy. No energy is required. No energy is required. Are you following, children? Understood? Or any doubt still here? We were telling diazotrophy means nitrogen fixation. What is nitrogen fixation? Conversion of nitrogen to ammonia. Reduction of nitrogen to ammonia. Who will do it? Nitrogenase enzyme will do it. What is nitrogenase? Made up of 2 components. What equation it will do? Altogether, if you see the, altogether if you see the complete equation, the equation goes like N2. Now, how many protons are required here? 6 protons and 6 electrons, no, CH3. So, 2 ammonia molecules. N2 gets converted to 2 ammonia molecules. So, N2 plus we can give 8 electrons plus 8 protons. Nitrogen combines with 8 electrons and 8 protons. Each electron requires 1 ATP. Each proton also requires 1 ATP. Means 8 ATP here, 8 ATP here. So I'm clubbing, I'm telling 16 ATP. 8 plus 8, 8 2s are 16. So 16 ATP molecules are required. Means nitrogen fixation is not a simple process. It requires too much amount of energy. 16 ATP molecules are required to reduce N2 molecule into 2 molecules of ammonia. To reduce N2 into 2 molecules of ammonia. 2 3s are 6. but we added 8 hydrogens and 8 electrons. What happens to the extra 2 hydrogens? They come out as H2. So H2 plus 6. 6 plus 2, 8 got over. Now, we told that hydrolysis of ATP, energy is consumed. Means 16 ATP is broken down to 16 ADP plus 16 PI. ATP is hydrolyzed to ADP and PI. This is a complete process of nitrogen fixation done by the enzyme nitrogenase. Done by the enzyme nitrogenase. So, we should be very careful about this equation, because from this equation 2019 NEET also a question came. 2020 NEET also question came. 2020. Now what was the question they asked in 2019? They asked for one ammonia molecule how many ATPs are consumed. For one ammonia. How many ATP is a question, children? Now come on, check and tell me. This equation what you wrote is for 2 ammonia molecules. For 2 ammonia molecules, 16 ATP. In the question they asked you for 1 ammonia. That means 16 by 2, 8 ATP. For 1 ammonia, how many ATPs means? We got the answer. What is it? 8 ATP, right? The answer is 8 ATP. If you don't understand the concept, if you see 16 ATP, you can mark 16 ATP and come. It is absolutely wrong, because they are not asking you for 2 ammonia molecules. They asked you for 1 ammonia molecule, isn't it? NEET 2020, what question they asked? In NEET 2020, they asked what are the products? What are the products of nitrogen fixation? This is the question. What are the products of nitrogen fixation? Now these are the substrates and they are the products. These are the substrates and they are the products. Now what are the products they asked? First option they have given ammonia only. First option. Second option they have given ammonia and hydrogen they have given. Third option they have given nitrite. Fourth option they have given nitrate. What is the answer? Tell me. What are the products of nitrogen fixation? The products are ammonia with hydrogen. That is the answer. NEET 2020, this was the question. Ammonia plus hydrogens are the products. Most of the children made mistake here and they just took this equation into mind. So they told only ammonia is formed, but when you balance the equation, this is a complete process, right? So ammonia with hydrogen are the products of nitrogen fixation. This equation is there in the NCERT textbook, but this equation is also there. This equation is also there. So in this equation, in these options, they told ammonia only. That only will make mistake, right? It's not only ammonia. Ammonia with hydrogen also. Take a screenshot. We will continue. Right, children? Let's continue with nitrogen fixation only. We understood about the enzyme, right? The enzyme contains iron also. The enzyme contains molybdenum also, right? And for reducing the nitrogen to ammonia, we require 8 electrons and 8 protons, isn't it? We wrote the equation, 8 electrons and 8 protons equation we wrote, right? Now, N2 plus 8 electrons plus 8 protons plus 16 ATP gives rise to 2 ammonia molecules plus H2 plus 16 ADP plus 16 PI. So this is an equation. Now the question here is, from where are you getting these 8 electrons and 8 protons, correct? From where are we getting the 8 electrons and 8 protons? Now, do you remember glycolysis? Do you remember glycolysis? We start with glucose. Glucose is broken down to pyruvic acid. Glucose is broken down to pyruvic acid. So the end product of glycolysis is pyruvic acid. Now pyruvic acid gets converted to acetyl-CoA and enters the Krebs cycle, we will tell. Pyruvic acid gets converted to acetyl-CoA and enters the Krebs cycle. From there only we will get electrons and protons, right? So we can tell that the electrons and protons. The electrons and protons required for reduction of N2 to ammonia. From where do they come? They come from pyruvic acid. They come from pyruvic acid. And what is pyruvic acid? Pyruvic acid is the end product of glycolysis. When you break glucose, you will get pyruvic acid. So the electrons and protons required for the reduction of nitrogen to ammonia comes from pyruvic acid. What is pyruvic acid, Emma? It is the end product of glycolysis. It is the end product of glycolysis, and the equation is called as, the reaction is called as phosphoroclastic cleavage. What is that reaction called? It is called phosphoroclastic cleavage. The reaction name is called phosphoroclastic cleavage. Follow carefully. Glucose, which is a 6-carbon compound, it breaks down to a 3-carbon acid. The name of the 3-carbon acid is pyruvic acid. Now start with pyruvic acid, which is a 3-carbon compound. Pyruvic acid, which is a 3-carbon compound, when it undergoes cleavage. Phosphoroclastic cleavage in the presence of ATP. ATP gets converted to ADP. Then what happens? The 3-carbon compound is breaking down to acetyl phosphate and carbon dioxide. Acetyl means 2 carbons. 2 here, 1 here. 2 plus 1, 3. Pyruvic acid. So pyruvic acid uses ATP and gets converted to a 2-carbon compound called acetyl phosphate and a carbon dioxide molecule. Now, in this process, it loses electrons, it loses protons. In this process, when pyruvic acid is getting converted to acetyl-CoA, it will lose electrons and it will lose protons. Now, electrons are accepted by what? From this reaction to this reaction, electrons are lost. Electrons are accepted by ferredoxin. Electrons are accepted by ferredoxin. Whereas protons are directly accepted by nitrogenase enzyme. While protons are directly accepted. Protons are directly accepted by, protons are directly accepted by nitrogenase enzyme. Electrons are accepted by ferredoxin, while protons are directly accepted by nitrogenase enzyme. Got it, children? Understood? Now, the electrons are first accepted by ferredoxin. Ferredoxin will give to component to dimer iron protein. Ferredoxin will give to iron protein. Iron protein will give to iron molybdenum protein. Iron molybdenum protein will give to nitrogen. And then the nitrogen will take protons also, will take electrons also, becomes ammonia. Will take protons also, will take electrons also, becomes ammonia. This is the process, children. So what did I write here on the board? Try to understand. We took the complete equation of nitrogen fixation. N2 with 8 electrons, 8 protons. For each electron, 1 ATP. For each proton, 1 ATP. Total 16 ATP will give you 2 ammonia molecules. 2 hydrogens which are left out will come. ATP is hydrolyzed, is broken into ADP and PI. Right? Now, for doing this, it's clear that we need electrons and protons. The electrons, protons required for reduction of nitrogen to ammonia, from where do they come? When glucose is converted to pyruvic acid. What is pyruvic acid? The end product of glycolysis. From there it will come. What is the reaction called? Phosphoroclastic cleavage. How does it occur? Pyruvic acid, which is a 3-carbon acid, When it takes ATP, it breaks into 2 carbon compound, 1 carbon compound. 2 carbon compound is called acetyl phosphate. 1 carbon compound is called carbon dioxide. Then it loses electrons and protons. Electrons are taken by ferredoxin. Protons will directly taken by the enzyme. So electrons are taken by ferredoxin. Ferredoxin will give to component 2, no? It will give to component 2. Component 2 will give to component 1. And component 1 will give to nitrogen. So this is the equation. Take a screenshot. We will continue. Right. So I'll try to explain it in a little detailed manner. Right. We will see the box equation. Then I think you can understand it. So, ferredoxin. Ferredoxin is reduced form. So, it took the electrons, right? After taking the electrons, ferredoxin gets reduced. When ferredoxin becomes oxidized, it will pass on the electrons to component 2. And then comes the component 1. Summa, what is this? This is component 2 of nitrogenase enzyme. What is this? Component 1. Component 2 is called dimer. Component 1 is called tetramer. Now, so dimer contains 2 iron molecules, right? Here we have 1 iron, here we have another iron. Component 1 is a tetramer. It contains iron molybdenum and again iron molybdenum, right? Now, when reduced one, after taking electrons, ferredoxin got reduced. So, when it is getting oxidized, it will give the electrons to iron. It's giving the electrons to iron. Now, iron was oxidized. After taking the electrons, iron becomes reduced. Iron want to get back oxidized. When iron is getting back oxidized, it will pass on the electrons. It will pass on the electrons to iron molybdenum. Iron molybdenum, it is oxidized. After taking the electrons from here, it gets reduced. Iron molybdenum want to get back oxidized. This is what is called redox reactions. So they are reduced, they get oxidized. So then they give it to the next one, the oxidized one gets reduced. Like that it go. Now iron molybdenum, which is reduced one, get back oxidized by passing on the electrons to nitrogen. Nitrogen has already taken hydrogen. So nitrogen plus hydrogen, protons plus electrons will give 2 molecules of ammonia. It will give 2 molecules of ammonia. And what is this component 2 and component 1? This is nothing but nitrogenase enzyme, right? And then we told the dimer works with the hydrolysis of ATP. At this step, children, 16 ATP will come. At the dimer, 16 ATP will come and they are hydrolyzed to 16 ADP and 16 PI. It is broken down to 16 ADP and 16 PI. Hope you understood this reaction in little detailed manner. In NCERT, they have given another diagram also. That also we will see. In NCERT, they showed the nitrogenase enzyme like this. This is a nitrogenase enzyme and these are the active sites. What are these? So these are active sites. Okay. Now, who will come to the enzyme? N2, right? Triple bond is there. So nitrogen is coming. N2 is coming and it will occupy the active site. Right? See? Now, N2 came and occupied the enzyme. What is this called? Enzyme substrate complex. What is this called? This is called enzyme substrate complex is formed. enzyme substrate complex is formed. Now we will do reduction, right? So we will add 2 hydrogens. So if you add, what is this step called? Reduction. So when you add 2 hydrogens, one triple bond becomes double bond. Let us see what happens. N double bond N, N double bond N, and one bond got broken, the hydrogens will get added. Okay. One hydrogen is added here, one hydrogen is added here. It becomes NH, NH. So I'll write reduction here. Now, this is free enzyme. This is enzyme with the substrate. What is this called? This is called diamide. What is this called? Right? Imide. Now, next again, what happens? 2 more protons, 2 more hydrogens are added. Then again the reaction is called reduction. Again the reaction is called reduction. Now this time what happens? One more bond will break. 2 more hydrogens are added, right? It becomes N single bond N. N single bond N. When these 2 hydrogens are added, so what happens? One hydrogen is here, another hydrogen is here. One hydrogen is here, another hydrogen is here. So what is this called? NH2, NH2, hydrazine. What is this called? This is called hydrazine. And children, again 2 more hydrogens, means one more reduction. One more reduction. Now this bond also will break. The single bond also will break. Then what happens? N and N. Bond is not there. Bond is not there. Then all the 3 hydrogens can bind. So 1, 2, 3. 1, 2, 3. First hydrogen, second hydrogen, third hydrogen. First hydrogen, second hydrogen, third hydrogen. 2 ammonia molecules are formed. 2 ammonia molecules are formed, right? So first reduction, second reduction, third reduction. In first reduction, diamide is formed. In second reduction, hydrazine is formed. In third reduction, 2 ammonia molecules are formed. Now what happens? These 2 ammonia molecules, right? These 2 are the ammonia molecules will be released out. Let me show that. Two ammonia molecules are released out. Right? So 1, 2, 3, NH3. 1, 2, 3. So 2 ammonia molecules are released out. And what happens to the enzyme? So after these 2 ammonia molecules are released out, the enzyme is free again. The enzyme is free again. Enzyme straight away coming to its place. Enzyme straight away coming to its place to do one more reaction. To do one more reaction. Now check how many hydrogens are consumed. 2 plus 2 plus 2. 2, 4, 6. But how many did you take here? 8. In the previous reaction, we wrote 8 electrons and 8 protons. Now 6 only we need, right? 2, 4, 6. Why 8 came? So actually, step by step hydrogens are coming. So they don't know that the process got over, and 2 more hydrogens will get added here. 2 more hydrogens will get added here, but where can they occupy? Nitrogen valence is 3 only. It can take max of 3. So that is why those 2 hydrogens will be released out as H2. Those 2 hydrogens will be released out as H2. Children understood? We completed nitrogen fixation in utmost detailed manner. In very detailed manner, we completed nitrogen fixation. Now, the nitrogen fixation mechanism, biological nitrogen fixation, is also called diazotrophy, mediated by an enzyme called nitrogenase. It has 2 components. Component 2 dimer, component 1 tetramer. Dimer contains 2 iron molecules, tetramer contains iron molybdenum and iron molybdenum. The equation is N2 plus 8 electrons plus 8 protons. From where are the electrons coming? From pyruvic acid, right? Who will take the electrons? Ferredoxin. After taking the electrons, it gets reduced. It will get oxidized, pass on the electrons to iron. Iron passes on the electrons to iron molybdenum. Iron molybdenum pass on the electrons to nitrogen. Nitrogen takes protons and electrons, it becomes ammonia. In another manner, they represented this equation where this is a nitrogenase free enzyme, ammonia N2K men bound, enzyme substrate complex, 2 hydrogens, one covalent bond is broken, it became diamide. 2 more hydrogens, 1 more covalent bond is broken, hydrazine. 2 more hydrogens, that single bond is also broken, 2 ammonia molecules are formed, and 2 hydrogens are still on the way, they came, but nitrogen is telling no, my valence is fulfilled, I can't do anything, they will go out. And 2 ammonia molecules are released and the free enzyme is released. The enzyme is coming back and it's ready to do the next step, children. Understood? So nitrogen fixation, this is nitrogen fixation. So I will write here the nitrogen fixation in plant is controlled by a gene called NOD gene. Nitrogen fixation in plant is controlled by NOD gene. And nitrogen fixation bacteria is doing, no? Nitrogen fixation in bacteria is under the control of 3 genes. Bacteria also has NOD genes, it also has NIF genes, it also has fix genes. NOD genes, nodulating genes. NIF genes, nitrogen fixing genes. NIF genes are nitrogenase genes and fix genes are nitrogen fixing genes, right? So this point is also important. In plant only NOD genes are there, whereas in bacteria NOD genes are there, NIF genes are there and fix genes are there. Now we completed the first step. What is that? Nitrogen fixation. In the last lecture, we'll try to complete the remaining steps called nitrification, nitrogen assimilation, ammonification and denitrification. If you like the content, share the link or share the channel with your friends. Thank you.